If you've ever opened a research paper and thought, I want to understand this, but this is dense, then this podcast is for you. Every day, thousands of research papers are published.
Most of them are rigorous, insightful, and almost impossible for non-specialists to read comfortably. But the ideas inside them shape medicine, technology, business, and society.
Welcome to One Paper Orbit. We are a research paper podcast. Here, we take peer-reviewed research and turn it into clear, structured conversations without adding speculation, without exaggeration, simply making the original work easier to understand.
Today, we are discussing a paper authored by Aida Kamalian, Polina Shishkova, Marco Tomietti, Christopher Below, Sarah G. Ho, Roland Bruderer, Lucas Reiter, Yuhan Feng, Michael W. Lutz, and Apai Mugeker, titled Unveiling Proteomic and Peptide-Level Modifications in Cerebrospinal Fluid and Plasma in Normal Cognitive Aging, published on 18 November 2025 in Communications Medicine.
This discussion is based on a paper published under the Creative Commons Attribution 4.0 license. The original authors retain credit for their work.
This podcast provides an adapted explanation for educational purposes, with full citation and license information provided in the description.
Okay, let's unpack this because, usually when we hear about neuroscience or like brain studies on aging, the focus is almost exclusively on disease.
Right, exactly.
We talk about Alzheimer's, we talk about dementia, what goes wrong. Science typically studies the brain after the house is already burned down.
Yeah, we spend all our time looking at the ashes, right? Trying to reverse engineer the fire.
Exactly. But the mission of this deep dive is completely different. Here, we are doing a highly detailed home inspection on a perfectly functional, older house, just to see how the wood naturally settles over 40 years.
Which is so important.
Right. We are trying to establish the absolute baseline of a cognitively normal, aging brain.
And what's fascinating here is you cannot truly understand a disease state until you have a flawless map of that healthy baseline.
Makes sense.
And historically, defining a healthy aging brain at a molecular level has been, well, incredibly difficult because of the sheer noise in the data.
So much static.
Exactly. But these researchers bypassed that noise with a very specific, rigorous study design. They looked at 92 cognitively normal individuals, and they split them between a younger cohort with a mean age of 29 and an older cohort with a mean age of 69.
And just to clarify, these individuals had totally normal clinical dementia ratings.
Yes, normal ratings, normal MRI scans. But the real breakthrough is how they sampled them. They took cerebrospinal fluid, or CSF, and blood plasma simultaneously from the same fasting individuals.
Which minimizes individual variability. I mean, if you compare my blood to your brain fluid, the genetic and lifestyle differences between us create so much static that it's hard to see the actual age-related changes.
Right, it's comparing apples and oranges at that point.
Exactly. By taking matched samples at the exact same moment from the exact same person, we get to observe the real-time crosstalk between the peripheral body and the central nervous system.
It allows us to track what is actually moving across the borders, but, having the right samples is only half the battle.
Correct.
They analyzed these fluids using liquid chromatography with FAMES. That's field asymmetric ion mobility spectrometry.
Okay, that is a mouthful. What does that actually mean for the data?
Well, traditional mass spectrometry often struggles with isobaric peptides. These are molecules that have the exact same mass but different three-dimensional shapes.
Oh, okay.
So FAMES applies an alternating high-voltage electric field to a gas phase. As the peptides drift through this gas, their physical shape and charge dictate how they tumble and maneuver through the electric field.
Wow.
Yeah, it essentially sorts the molecules by their aerodynamic drag at a sub-microscopic level.
Okay, let me make sure I'm visualizing this correctly. Standard proteomics might just tell us how much of a specific protein is floating around in the fluid, right? It just weighs the material.
Essentially, yes.
But by using FAMES, the researchers can see the physical folding, the structure, and the exact physical condition of the proteins. It's like knowing not just that there are bricks in the fluid, but whether those bricks are, whole, chipped, or painted.
That structural insight is the entire core of the paper. When they applied this high-resolution sorting to the macro level, looking at whole proteins, the differences between the 29-year-olds and the 69-year-olds were stark.
What did they find?
In the blood plasma, the most notable shift was a significant downregulation of the IGF-1, or insulin-like growth factor, signaling pathway.
Okay, IGF-1.
Right. This is a highly conserved pathway linked to organismal lifespan across multiple species. As the body ages, it basically dials down this growth signaling.
I follow the logic there. The body is no longer in a growth phase, so it scales back. But the findings in the cerebrospinal fluid are where I really need to push back, because they seem entirely paradoxical.
Oh, they definitely do at first glance.
Right, because the researchers saw a massive upregulation in coagulation pathways, like 3 specific fibrinogen domains, which are heavily involved in blood clotting, jumped by 40% in the older adults' CSF.
Compared to just a 12% increase in their blood.
Right. And alongside that, they recorded a huge spike in neuroinflammatory pathways and complement activation cascade members. So if these older adults are cognitively normal and their brains are ostensibly healthy, why is their central nervous system flooded with clotting factors and immune system attack cascades?
This is a great question.
It sounds like a biological war zone.
If we connect this to the bigger picture, it only looks like a war zone if you view inflammation strictly as a destructive force.
Way, isn't it?
Not always. Gerontologists refer to this chronic low-grade increase in inflammatory markers as inflammaging.
Inflammaging. I like that term.
Yeah, but in the context of a healthy, cognitively intact older brain, this isn't necessarily a failure state. The researchers found a massive remodeling of the extracellular matrix occurring simultaneously.
The extracellular matrix, that's the structural scaffolding around our cells, right?
Exactly. In these older brains, core matrix glycoproteins increased dramatically, while various collagens actually decreased. So the physical environment is being actively restructured.
Are you suggesting the brain is purposefully changing its own scaffolding to accommodate the inflammation?
The hypothesis presented in the paper goes even further than that. The extracellular matrix remodeling and even the localized inflammation might actually be a protective adaptation.
Wait, really? Protective?
Yeah. Think about neurodegenerative states like Alzheimer's. Harmful, misfolded proteins like phosphorylated tau or amyloid beta propagate and spread through the brain tissue.
Right, the plaques and tangles.
Exactly. The researchers suggest that in healthy aging, the brain proactively alters its extracellular nets. It changes the composition of the glycoproteins to create a denser, more reactive matrix designed to catch, sequester, and inhibit those harmful proteins from spreading.
Oh, wow. So the inflammation in the scaffolding changes aren't the house burning down. It's the brain actively building fire breaks.
That's a perfect way to put it.
It's installing a denser structural net to trap the ambient toxic proteins that, naturally accumulate over time.
Yeah.
That drastically changes how we should interpret high inflammation in a healthy older patient.
It forces us to reconsider what we call dysfunction versus what is actually a mature defense mechanism.
That is mind-blowing.
It really is. But to understand how the brain builds these fire breaks, we have to transition from the macro level of whole proteins down to the micro view, the peptidomics, and the post-translational modifications.
Oh, okay. The peptides.
Right. This is where the FAMES technology we discussed earlier actually pays off. The researchers weren't just looking for protein abundance, they mapped out alternative cleavage.
Which is where a protein is physically cut, right?
Yes, exactly. And phosphorylation, where a negatively charged phosphate group is chemically bonded to the protein.
Here's where it gets really interesting. And I want to use an analogy because the concept of post-translational modifications can get a bit buried in the biochemistry.
Go for it.
Imagine using a drone to survey a parking lot. Traditional proteomics is like the drone flying high above and reporting back that there are 50 Honda Civics in the young parking lot and 50 Honda Civics in the old parking lot.
Right.
So a standard antibody test would conclude that both lots are identical because the total count is the same.
Which completely misses the functional reality on the ground.
Exactly. The peptidomic approach in this study is like sending a mechanic down to walk the lot. The mechanic reports back that yes, there are 50 Civics in the older lot, but 20 of them have had their steering wheels stolen, and 10 of them have a brand new custom paint job.
I love that.
Right. They are technically the same cars, but they do not drive the same. The molecular modifications fundamentally alter the utility of the vehicle.
And, the APOE protein is a perfect example of this mechanical shift.
Oh, APOE, that's a big one in Alzheimer's research.
Huge. APOE is critical for lipid metabolism. It processes fats and cholesterol in the brain. At the whole protein level, the drone survey showed normal amounts of APOE in the healthy older adults.
But the mechanics saw something different.
Completely different. The peptidomic data revealed that in the older cohort, the APOE protein was experiencing a high rate of cleavage, specifically at amino acid 144.
Okay, let's break down the mechanics of that specific cut. Why does clipping the protein at position 144 matter so much?
Because position 144 is located in the physical hinge region, it connects the lipid-binding tail of the protein to the receptor-binding head.
Oh, wow.
Right. APOE's job is to grab a lipid and then bind to an LDL receptor to deliver it. By cleaving the protein right at that hinge, you physically uncouple the payload from the delivery system.
So the protein is technically still floating in the fluid,
Yes.
but it can no longer fold correctly to bridge the lipid to the receptor. Its primary metabolic function is severely compromised, not because the protein is missing, but because its structural integrity has been severed by the normal aging process.
Exactly.
That's a staggering realization. The body is still spending the energy to produce the APOE, but the ambient environment of the older brain is functionally neutralizing it by clipping that hinge.
It's wild, and the study found a highly specific modification with the amyloid precursor protein, or APP as well.
APP. That's the notorious protein that, when processed incorrectly, forms the amyloid plaques associated with Alzheimer's, right?
Yeah, that's the one. And this APP finding is perhaps one of the most vital insights in the entire paper. In disease states, APP is cleaved by beta secretase enzymes at specific sites, creating those toxic fibrils.
Got it.
But in these healthy, sharp, older adults, the cleavage was shifted to a completely different location, AS142. Yeah. And this specific amino acid sits inside a highly preserved copper binding domain.
Wait, if the cleavage happens at the copper binding domain, how does that physically change the life cycle of APP? I mean, copper is just a trace metal. Does it actually dictate where the protein goes?
It dictates exactly where the protein goes.
Really?
Yeah. The binding of copper to APP modulates how the protein dimerizes, how it pairs up with other molecules, and that pairing determines its subcellular sorting. It acts like a routing tag for the cell's transport machinery.
Okay, I see where you're going with this.
By cleaving ATP at the copper binding site, the healthy aging brain is physically removing that routing tag. This changes how APP moves through the cell, likely keeping it out of the specific endosomal compartments where the toxic beta secretase enzymes live.
So by clipping off the copper binding domain, the aging brain is effectively rerouting the traffic.
Exactly.
It forces APP down a safe, non-amyloidogenic pathway. It's intentionally disabling the protein's ability to be turned into a plaque.
It demonstrates that APP has a dynamic, normal life cycle that actively shifts as we age. It isn't just a static molecule waiting to become a disease.
Right.
The healthy brain actively adapts its processing to avoid that outcome.
Okay, so we have these floating proteins like APOE and APP being rerouted and clipped. But if we scale back up slightly from the individual peptides to the actual physical tissue of the brain, like the synapses and the blood vessels, how do these microscopic cuts impact structural integrity? Are the neurons themselves pulling apart?
The data suggests they are certainly loosening.
Loosening.
Yeah. The researchers examined a synaptic hub protein called NRXN1.
NRXN1.
Right. This is a transmembrane protein that acts as a structural tether. It reaches across the synaptic cleft and, the tiny gap between neurons, and physically hooks onto proteins on the other side to hold the neural connection stable.
It's the literal glue maintaining the physical infrastructure of our neural networks.
Exactly. And in the older cohort, the researchers found increased cleavage of NRXN1 specifically at the LG4 domain.
And what is the LG4 domain?
The LG4 domain is a large L-shaped physical hook on the extracellular side of the protein. It is the exact molecular geometry required to interlock with the adjacent neuron.
Oh, no.
Yeah. When you cleave the protein at the LG4 domain, you sever the transsynaptic bridge. The neuron survives, the synapse still exists, but the physical tethering is compromised. The structural adhesion basically drops.
Which perfectly explains why cognitive processing speed or fluid intelligence naturally slows down as we age,
Right.
even if there is absolutely zero neurodegeneration.
Precisely.
The neurons are healthy, but the physical bridges holding the network tightly together are slowly losing their hooks.
Right.
But the finding that completely recontextualized this entire study for me was the data on the blood-brain barrier.
Ah, yes, the COL4A2 protein.
Right. They isolated a modification on COL4A2. What does that protein actually do?
So COL4A2 is a critical subunit of collagen IV, and collagen IV is the primary structural polymer that forms the basement membranes of the blood-brain barrier. It creates a dense, cross-linked mesh that prevents large molecules in the blood from seeping into the delicate environment of the brain tissue.
And what modification did the FAME's mass spectrometry find on this border wall?
A very specific phosphorylation event. A phosphate group was added to the SIR-1475 residue of the COL4A2 protein.
Okay, let's explore the biophysics of that. Because a phosphate group isn't just a passive tag, right? It carries a massive, bulky, negative charge.
Exactly.
If you take a tightly woven structural mesh like collagen IV and suddenly introduce highly negatively charged phosphate groups into the lattice, what happens to the physical structure?
Electrostatic repulsion.
Oh,
The negative charges physically repel each other and the surrounding molecular structures. You are forcing a bulky, charged wedge into a tightly packed collagen triple helix.
that doesn't sound good.
It prevents the collagen protomers from assembling into their normal, dense hexamer networks. The threads of the basement membrane literally push themselves apart, causing the tight junctions of the endothelial cells to slightly loosen.
So what does this all mean? What does this mean for the physical blood-brain barrier inside your head? Are we saying that even if you stay perfectly healthy and sharp, your brain's border security fundamentally gets leakier?
That's exactly what the data implies.
Which brings us all the way back to the macro findings we discussed at the very beginning.
Yes.
If electrostatic repulsion is pushing the collagen threads apart and loosening the blood-brain barrier, that perfectly explains the fibrinogen paradox.
Right.
The massive 40% increase of blood-clotting fibrinogen in the brain fluid isn't because the brain is suddenly manufacturing it. The fibrinogen is leaking in from the blood because the barrier has become porous due to a single phosphorylated serine residue.
The cascade of causality is beautifully clear here.
It really is.
The targeted phosphorylation of COL4A2 introduces electrostatic repulsion. The barrier loosens. Large peripheral proteins like fibrinogen leak from the plasma into the cerebrospinal fluid.
And then the local immune system detects it.
Exactly. The local immune system of the brain detects these foreign blood proteins and triggers the complement activation pathways. This localized inflammation prompts the extracellular matrix to remodel its glycoproteins into denser nets to sequester the debris.
It all stems from a microscopic chemical charge altering the architecture of a single collagen thread. It is a stunning mechanical chain reaction.
Truly.
So, looking at the absolute biggest picture here, we have tracked the healthy aging brain from fluid crosstalk down to the specific peptide cuts that uncouple lipid delivery, reroute amyloid precursors, sever synaptic hooks, and electrostatically repel the blood-brain barrier.
We covered a lot of ground.
We really have.
Yeah.
What is the overarching clinical takeaway for the future of medicine? Why should you care about this?
The takeaway is that normal cognitive aging is a highly active, dynamic remodeling process. It is not a passive decay.
Right.
The brain is constantly adjusting its molecular architecture to manage the physical realities of time. If we misinterpret these structural adaptations as strictly pathological, our interventions will just fail.
And that is the danger of the current longevity mindset. Right now, immense resources are poured into therapies that aim to blindly reverse aging or, eliminate inflammation entirely.
Right.
But this paper proves that some of that aging, specifically the inflammaging and the extracellular matrix nets, might actually be your brain's customized defense system.
Yes.
If you deploy a blunt medical instrument to stop that inflammation, you might accidentally tear down the barricades your brain just spent 40 years building to trap toxic proteins.
Future therapeutics will have to pivot away from those blunt instruments. It will no longer be sufficient to just boost the overall expression of a protein.
Right, like we were saying earlier.
Administering a drug to flood the brain with more APOE is completely useless if the ambient environment is just going to cleave the hinge at amino acid 144 anyway.
You just end up with more broken delivery trucks in the parking lot.
Exactly. The therapies of tomorrow will be precision structural medicine. Pharmacologists might design small molecules that act as chemical shields, binding specifically to the hinge region of APOE to physically block the enzymes from cutting it. Or they might develop localized phosphatase inhibitors that specifically remove that negatively charged phosphate group from COL4A2.
And that would allow the collagen mesh to pull tightly back together and seal the blood-brain barrier.
Exactly, without disrupting the protective matrix nets nearby.
It requires a level of mechanical precision that respects the complexity of the system rather than just trying to override it.
Absolutely.
Which leaves us with a truly provocative thought to consider. If a healthy, cognitively sharp older brain relies on this intricate symphony of active structural changes, cleavage, and rerouting, Maybe the holy grail of neuroscience shouldn't be stopping aging entirely.
Right.
What if the secret to preventing neurodegeneration isn't aggressively keeping the brain completely young, but rather providing the specific structural support our proteins need to adapt gracefully?
That's a fascinating way to look at it.
What if the goal of medicine shouldn't be freezing the house in time, but simply helping the foundation settle without cracking?
We have to learn to support the architecture of age rather than trying to erase it.
This paper is published under a Creative Commons Attribution 4.0 license, and full credit is provided in the episode description. Thank you for listening to One Paper Orbit Say, our research paper podcast. Our goal is to make peer-reviewed research accessible to everyone. If this episode was helpful, please share your thoughts and let us know which papers you'd like us to explore next. Thank you.